cell biology. Here are a couple of reviews that we wrote. They're still pretty current, but I'm going to focus on the genetics. Why are you all in genetics? What's the difference between genetics and all other branches of biomedical science, like biochemistry, cell biology? We'll just start with Maya. Maya or Maya?
Maya.
Maya. What's better about genetics? Why are you guys all so smart for choosing genetics?
I guess part of the reason I chose it is that I think it's a lot of focus on like how things are regulated and how that kind of can like intersect with like disease and not just like it's a lot of like mechanism, but also like thinking about what enables the mechanism to happen and at what time it is.
Okay, that's one good reason. How about you?
I think it's basically based on theoretical concepts. I mean, not before the era of molecular genetics. I think it's basically theoretical studies.
Anyone else? Those are good reasons. Anyone else have a particular reason they chose genetics? Let me tell you mine. It's the only discipline you can make firm conclusions on. Because you can't prove anything in science. All you can do is get evidence to support a hypothesis. And the evidence that you can get to support a hypothesis can be quantified with statistics in any discipline, but only in genetics in the biomedical research disciplines can you really get probabilities that are actual and validatable. For instance, with functional studies, we'll talk about tumor suppressor genes and oncogenes a lot. Let's say you try to do a functional study that shows some gene is an oncogene. And you inject the gene into a cell and find out that it behaves like a cancer cell should. What does that mean? Carter, what does that mean?
That means there is, I think the cop-out answer is to say that means that that gene is sufficient for producing cancer. Although I don't know if we could say that gene behaves like other oncogenes in the sort of pathways that it induces cancer. I don't think we can say that yet.
Yeah, well, you can say anything you want, okay, in a paper, and people usually do, and they might say something like you said in the title. But that is just wrong, okay? It's not a cancer. Cancer is not something in a cell line that you can measure, number one. Number two, what are the controls? What are the probability? It really functions as an oncogene in human cancer. Those studies, they take a single gene in general. I'm not talking theoretical. There are literally hundreds of papers like this, and many of them are wrong. And they put in a gene and they see some phenotype. And it could be cell culture, it could be zebrafish, Drosophila mice. And then they conclude this gene is an oncogene. Now, what are the controls? They often have a control where they mutate the oncogene, and it doesn't work. Well, but whenever you mutate a gene, it might not fold properly, it may not be expressed, not really fully interpretable. And what you really should do is test every one of the 20,000 genes in the same system to see how different that gene is than the others. And additionally, even if that gene functions in that cell, the only conclusion you can legitimately make is that this particular gene, when injected in this particular cell, makes them grow differently. Whether that has anything to do with cancer or not is total speculation. Now think about genetics. When we look at genes, and putting a probability on that statement that is different than others, whenever you say something is different, you need to have a statistical evaluation, right? And I don't mean p-values. Again, I'm going to try in these two lectures to introduce you to concepts that usually aren't taught in classes, but I found are extremely useful for being a researcher. It's not just a p-value less than 0.05, whatever alpha is taken. That's just the beginning. The key is the effect size. That means how different it is than whatever the controls are. You can have a p-value of 0.05, let's say a drug for cancer, we'll keep it on cancer, that prolongs life by 1 week over a control. And that's statistically significant if you have enough patients, but it's not meaningful in a real clinical sense. What you would really like to show is that it increases survival by at least months, hopefully years. So it's not just statistical significance. Now, getting back to genetics, when we look at a gene, the only good way to find a driver gene, which we'll talk about in a few minutes, is it's the frequency with which it's mutated in cancers. And we look at a cancer, say breast cancer, doesn't matter, and we find that a given gene is mutated in a certain way 1,000 times more than any of the other 20,000 genes in the human genome. You can define a probability associated with that, a p-value which will be something like 10 to the minus 20th, and the effect size is huge. Then you know it's an oncogene, or a driver gene. You can tell whether it's an oncogene or a tumor suppressor gene by the nature of mutations, which we'll talk about. But there have been many papers which have mistakenly identified genes as cancer driver genes, which aren't. The number of cancer driver genes is just a few hundred. How many genes are there in the human genome? Lauren?
20,000?
How many? Louder. Doesn't matter. Don't worry, we're going to get these answers right
Okay.
along. Okay.
20,000.
How many?
20,000?
20,000. Anybody go higher or lower? Okay. 20,000 it must be. Haley, what's a gene?
A region of the DNA that encodes for a protein.
It has to confer protein, it has to...
I guess that's the classical definition.
Classical, so what is it now in 2025?
Could be involved in pathways that are important for cellular function.
Yeah, but what can they code that aren't proteins that are important?
Like long non-coding RNAs or
Non-coding RNAs,
Yeah.
siRNAs,
yeah.
right? Ribosomal RNAs, transfer RNAs. Are there any cancer genes which don't encode proteins? Valeria, that have so far been defined?
I have no idea.
Good answer. Okay. is no.
Okay.
Dema or Dena?
Dina.
Gina. Okay, please excuse my pronunciation. Why haven't any non-coding genes been found to cause cancer? Just imagine, I know you don't know the answer to this question.
Maybe people just haven't like looked in that direction.
Great answer. There are two possibilities. People haven't looked at them enough, and that used to be true. But the other answer is they just don't function as cancer driver genes unless they alter the coding sequence. How about if they alter the promoters? So they're coding genes in the sense that they code for proteins, Anthony, but they don't, the mutations themselves do not alter the amino acid sequence, right? So they don't cause amino acid changes.
So a silent mutation not affecting the function of a...
Well, no, they
A promoter.
could be
Oh,
a
okay.
promoter,
Oh,
an
a promoter.
enhancer, polyethylation site, 5' UTR, 3' UTR, enhancer. Let's make it even general. You look at all human genetics, what fraction of mutations actually change the coding part of the gene? Not just cancer, but all human genetics. Take a guess. I don't expect any of you to know the answers to these, else I wouldn't have to teach you. Just guess. It's more fun to guess. It makes the answer more meaningful if you think about it for a few seconds before I tell you the answer.
So what percentage of the 2% of coding genes...
What percent? You take every patient who has an inherited mutation, forget cancer for a minute, okay? Cystic fibrosis, muscular dystrophy, doesn't matter, right? That's 100%. How many of those are due to mutations, inherited mutations, that don't affect the protein amino acid sequence, that are not missense mutations or splice site mutations within the coding region or nonsense mutations?
Probably less than 1%.
That's a reasonable answer. Anyone care to guess something different? Yes.
I'm gonna guess 50%, but only because I spent my whole last rotation looking at Fragile X syndrome. But biased because of that.
Yeah. So it's a as yet unanswered question, right? I mean, the only way you can tell is if you look at, say, a disease, let's just say cystic fibrosis, a classical disease, and you look at patients who have classical cystic fibrosis, and the way the assays are done now for hereditary predispositions is, at least up until recently, and still mostly practically, you just look at the exons of the gene, right? And if you see 90% of those patients have mutations in the coding region, then you can say that the other 10%, well, let's go around. And this will come up in our clinical discussion on Wednesday. Who's next? Zoe? Or Daniel?
Daniel.
Daniel. Okay, so it's 10%. You don't find it. What are the possible reasons for that?
Well, they could be in introns or in the enhancer regions instead.
Yep, those are good reasons. Zoe, what's another reason?
I guess you already talked about introns, but I guess like affecting splicing. So also be in introns, enhancer regions.
Yeah. And there's one other reason, which is a
Maybe.
cool one.
Oh, regions that are like,
Nope.
so,
Remember, there are other genes in the genome besides CF.
oh, like modifier genes?
Could be a no.
No.
Modifier gene usually work in conjunction,
Okay.
right? So they can modify the effect of the gene, but they themselves. We're talking about a monogenic disorder. Then it's usually a single gene, but maybe there's another gene that can cause CF that hadn't been discovered yet, right? So that's possible. It's not true for CF, but it's true for a lot of other diseases. CF is a true monogenic disease. I don't know of any mutations in genes other than the CF1 gene or whatever it is that causes cystic fibrosis. And yet there's another problem, the opposite problem. So let's say you're looking for a mutation in BRCA1 or BRCA2. And in a patient who's suspected to have hereditary predisposition to breast cancer. And now you're only looking at coding region. Oh, by the way, to finish that section, so whole genome sequencing is often what's done now. Or at least look at common mutations in introns, promoters, etc. There aren't many of them, but it depends on the disease. Another possibility is duplication of the gene that can cause problems in diagnosis. Also, a big problem is suppose the whole gene is deleted and you do exon sequencing. Or suppose a whole exon is deleted. What do you sequence? The only thing that's left is the normal copy. It looks normal, right? Your report back is totally normal. Wrong. Okay? So when and it's pretty complex, but there are ways to now look for duplications, deletions, and basically there are variations on whole genome sequencing or targeted sequencing. Eventually it'll all be whole genome sequencing. Nobody's going to do targeted sequencing. The price for doing a whole genome sequencing now, anybody know?
$500.
At
About
what X? What X coverage?
30.
30X. Yeah, that's about right. Clinically, because of stuff, it costs more. But the actual cost of goods for doing it is several $100. It's going to come down by 5-fold in the next 5 years. So, anyway, you get a mutation in a coding region, and whose turn is it? Janae? Janae. God. I should have written the phonetics spelling. I'll never remember this. Janae, so this just happened with a person I know well last week. So she gets the report, and it's a codon. It's a mutation at codon 957, which changes an arginine to a histidine. Okay? How do you report that to a patient? Well, you say you have an arginine to histidine mutation in codon 175, which doesn't mean much to the patient, or doesn't mean much to the patient's physician unless they're an expert in genetics. But how do you interpret that? Is that causing her disease or her predisposition to a disease?
I'm not sure. I guess it could. I don't know what
It
the
could,
charge
it's
difference
definitely.
between those two amino acids are. So I don't know.
Arginine to histidine is a big change. It's a very common mutation in general. But yes, it could. What do you think?
I mean, I might think about looking at, see if some variant databases have looked at lots of patients. Have they seen a higher incidence of cancer than expected with people who had a germline mutation of that kind? Is one idea, but there's probably a lot of other ways to do it.
Yeah, no, and that's a good idea, and that's precisely what's done. Okay, if you have a large database of people who have been sequenced for that gene, who are healthy, and now, there are literally hundreds of thousands of healthy individuals who have been sequenced with whole genome sequencing or exome sequencing, and you look in those to see what the incidence of that particular mutation in that gene is in healthy individuals versus those with breast cancer. And then you can assign a certain probability, statistically valid likelihood that mutation is pathogenic. Often, though, you don't find it in the database. You can also do functional studies, which are sometimes used at risk, again, because the functional studies are not nearly as good as the genetic studies, but that can increase your confidence legitimately. But still, so let's see, Alex, if I were to sequence your genome and also Carter's 500 hours a pop, it only cost me $1,000, much less in my lab. But how many differences are there between you and Carter? And now let's just keep it to single base substitutions. Okay? So we pretend your genomes are the same sequence read out, but now we're just looking at, A and T, A and C, A and G. How many changes? How different are you than Carter?
We have different backgrounds, so I don't know, maybe 1%.
How many differences? That's a percent difference. How many nucleotide differences? By the way, that 1% would be true if Alex were a grade 8. A grade 8 other than a human.
Oh.
You know what the difference between a grade ape and a monkey is? Anybody? Monkeys have tails. Next time you go to the zoo. All right, so yeah, if he were a non-human grade ape, it would be 1%, but it's less.
Is it 0.05%, like 2 million?
Closer, 0.1%. Okay, so in your haplogenome, and these numbers as geneticists, you should all remember 20,000 coding genes, number of base pairs in a cell. We'll start over. Maya, say it louder.
I want to say it's around 3 to the 9.
3 to the 9th, and always give me a unit. I come from a mathematical background, so I need a unit.
So
What is it?
3 to the 9.
Base pairs?
Yeah, it should be base pairs. And
Per?
Per cell or per diploid or
Per haploid.
per haploid.
So 6 billion per cell, 3 billion per haploid. Okay? So if it's 0.1%, you're different from from Carter in 3 million bases. That is the difference. Now, how else could he be different from you, Carter, besides single base substitution?
You might have some larger structural variants. Maybe I have some insertions that are, between 1 and 50 base pairs. It's also possible I think we were reading that a few percentage of people, like 1 to 2% of people have a significant copy number variation in the gene.
You're on the right track. Okay, structural variations, yes, they're often hard to define. There are relatively few of them, but they definitely exist, and everyone has them. Copy number variations is in everybody. It's not in a few individuals. Everyone has copy number variations. And Carter, your homework is to tell us next Wednesday, that's how we'll start, how many copy number variations there are in an average person and what their size is, the size of the duplication or deletion. Okay, and I'll give you a hand look at Evan Eichler's latest paper. He's done the best work on this. Okay, so you two differ in 3 million bases per haplogenome, which means you differ in 6 million bases per cell. How many of those bases, I'll pick a male, so we won't, introduce X and Y chromosomes. Who's the next male? Daniel. How many does he share of those 6 million? Actually, that's not a well-formulated question. I just want to introduce you to the fact that there are public polymorphisms and private polymorphisms. These variations in coding sequences, they're called polymorphisms. They're just single base substitutions. The difference between a polymorphism and a mutation is only functional. Polymorphisms generally refer to changes in genomes that don't cause disease, whereas mutations are associated with disease. But in general, I'd say now that so many human genes have been sequenced, or human genomes have been sequenced, about 2/3 of the polymorphisms are public. That means you'll find them in some percent of the population, say 1% of the population or 0.1%. The rest are private, which means you don't find them in any of the databases. Okay, that doesn't mean they're not in other people. It just means that you haven't sequenced enough people to see them, or they're rare. Okay, which means they just came up evolutionarily recently. The first incidence of that mutation occurred maybe a few hundred years ago for the really rare private mutations, or a few thousand years ago. Zoe, when did the first human appear on our planet?
Oh, I don't know.
Take a guess. Yeah, it doesn't matter. Don't worry about getting it right. Order of magnitude. 1,000, 10,000, 100,000, a million years ago.
Million?
No. A million below. It's 10 to the 5th. 100,000, something like that. Okay? Anthropologists will argue about that. But so the ones that are very common, right, occurred very early in that first few humans. And the recent ones occurred later. Now, the reason I mention all this is that when you look at these variants of unknown significance in the BRCA1 gene, when you see this mutation that's in somebody's BRCA1 gene but is not recorded in the database, you don't know if it's a private variant that is not in the databases but are pathogenic, were simply a harmless polymorphism that occurred in her great-great-great-great-great-great-grandfather. Okay, and I say it occurred in her great-great-great-great-great-grandfather rather than her great-great-great-great-great-grandmother. For what reason? Whose turn is it? Again, why is it more likely to have occurred in her paternal lineage in a man than a woman?
Greater offspring, like Genghis Khan kind of situation.
That's an interesting answer. But what wasn't the one I was looking at? Okay, let's look at it a different way. Dina, right? Okay. So how many mutations do you have that neither your mother nor father has? And your 6 billion base pairs in your genome.
Sorry. Obvious answer.
I think it's 100 in the exon. Is that like?
It's still in the right order of magnitude, okay? It's roughly 60, 70
Okay.
of the whole genome, though, not the exome. And how would you know that? Or how could you find that out for yourself if you wanted to?
Sequence myself and my parents.
Yep. And that's what's done. That's called trio sequencing.
Mhm.
Mom, dad, child. All right? So you find 60 or 70. Then you try to find out, and we're talking about constitutional. So they were present in the conceptus, the original cell that gave rise to you. Okay? How did that mutation get there? It was obviously occurred either in your mother's egg or your father's sperm. Which is more likely?
The father.
The father's, correct.
Mhm.
And Daniel?
Over here.
Oh,
I'm happy to
okay.
answer.
Wrong name. Yeah, so fathers, why?
They have a higher de novo mutation rate. I assume some of that's
Close.
okay.
You're on the right track.
And they have higher duplication. The spermatogenesis causes more splits over time.
Right, and this is also an important principle for genetics in general, not just cancer genetics. As we'll see, there are genes that predispose to cancer, just like those that predispose to cystic fibrosis. But it's easier for me to point. Malaria. What is the mutation rate in human cells?
Mutation rate?
Take a guess. It's good that you're thinking. That's what I want you to do.
What you don't see is that it's blank. Nothing's going on here.
You can phone a friend. They still have that show on TV. Phone a friend. What's it called?
Who wants to be a millionaire? Who wants to be a
Yeah,
millionaire?
yeah. Friend, you can ask the audience. Go ahead, phone a friend.
I think it's like 1 times 10 to the minus 8 for germline mutations and 1 times 10 to the minus 5 for C and Bs.
10 to the minus 8 for
Germline
for
mutation.
germline mutation.
Okay, I need a unit. That's miles per hour.
Base pair, no mutations. I don't know. Mutations per base in percent.
Yeah, that's close. It's mutations, and in the literature, when you're reading the literature, this is often confusing because they often don't give the unit. And it's very, and sometimes it can be very confusing. But anyway, the actual number is somewhere about 1 in a billion base pairs, 1 to 3, 1 mutation in, let's say, it's a little tricky, but you can remember a number per billion base pairs per cell division. Okay? That's true in all humans for all somatic cells. Okay? We'll talk about a somatic cell in a minute. So that means every time a cell divides, there are roughly 3 to 5 mutations that occur. That's, if you want to remember a single number, that's a good number. 3 to 5 mutations occur per cell division in a human cell. So the reason de novo mutations, that's what these mutations are called, that weren't inherited. They, about 70% of them occur in the male, in the father, and the remainder in the mother. And the reason is what you said, is because each sperm cell, each sperm goes through, I don't know, something like 25 or 30 divisions before it becomes, before its DNA is fixed in the sperm. I think it's actually 21, but it's a lot of division. Whereas the eggs don't go through that many. So, in females, your eggs, and I'm sure you already know this, your ova in females, they're done once you're in embryogenesis. You have no more division, none, they're fixed. Whereas sperm continue to reproduce throughout life. So you probably or heard that the older the father is, the more likely that the progeny will have a mutation, and that's the reason. The spermatic gonioid stem cells have divided more often. So it's not the mutation rate per se. It is the number of divisions. And this is very important in cancer. And we've got through exactly 0 slides. But I hope this is more fun than the slides. Anyway, it's very important because cancer, as we'll see, is a genetic disease. And what causes these mutations? Let's start that way this time. What's responsible for most mutations? No, I'll tell you, you look at a cancer, typical cancer, say pancreas cancer, colon cancer, has roughly 50 to 75 coding mutations, right? Another number you should remember, and by the way, you two guys, they're homework. You know, they get paid for doing this. You don't? Your work on
In a way, I guess. I get a stipend, so.
All right, so what I'd like you to organize, even though you're unpaid, is all the numbers that we've talked about in this session. Put them down on paper for Wednesday, and we'll have a slide we'll distribute it to everyone, because I think these are good numbers to remember. So, where was I? Okay, so what was I talking about?
50 to 75 mutations in a
50
cancer
to
cell line.
75 mutations. Yeah, so, and we were talking about the mutations in a cancer. 50 to 75 mutations in typical cancer. What caused those mutations?
It could be some sort of environmental insult,
Give me an example.
like UV radiation,
What kind of cancer?
skin cancer.
Good. Next. What else?
Like smoking could cause lung
Smoking,
cancer.
great example. Okay, so we have environmental causes. What else?
Error in
What we
correcting.
were just talking about. But hereditary,
Oh, okay.
right?
Yeah.
Those are mutations that are present that can contribute to cancer. As you'll see in a minute, whenever we talk about a multifactorial disease, we should not say cause. We should say contributor. Because no single mutation in this example is sufficient for cancer. It's always more than one. But anyway, so you have environmental causes, which are roughly responsible for about, in general, if you look at all cancers, 40% of mutations. 30 to 40. Pardon? Oh, okay. I just finished this thought. Thank you. That was good. What's every penny? 40%. Now, if it's a lung cancer, it's 90% because they're coming from smoke, right? If it's a skin cancer, it can be 90%. And that's why these environmental agents, this is how they contribute to cancer, by causing mutation. Hereditary mutations, it's just a few percent. Most patients who get cancer have no hereditary predisposition. And that's a very important principle. Now, some do, and that's also very important, because if you know who does, you can institute screening measures to catch the cancers particularly early and keep patients from dying. That's why all patients who have colon cancer now are tested for evidence that they might have a hereditary predisposition. And if they do, then their family members are tested. Similarly with breast cancer. A woman or a man has breast cancer, particularly a man, because breast cancers don't occur very much in men unless they have a predisposition. Then their family members are tested, and they, if the proband, that is the patient who started it all, has a mutation, is make sure they don't have the same mutation. If they do, then there are measures that are taken. Like for breast cancer, let's ask our wannabe MD, Maya. May or Maya? Maya. Maya, what would you advise your patient who is 20 years old and her cousin died or had a breast cancer? What should she do?
For the patient themselves, I would say they should probably have like earlier screening, either with ultrasound, probably because maybe ultrasound versus like mammogram.
Ultrasound for what?
Oh, like to see if if there are like any kind of like lumps that they kind of like feel on the same.
What cancers is she at risk for?
Oh, I would guess most likely like breast cancer, ovarian.
Yes, correct. Breast cancer and ovarian are the two main ones. Pancreatic cancer is the third one. In men, prostate cancer. But breast cancer, what's the risk? A typical patient who has a BRCA1 or BRCA2 mutation. What's the risk, lifetime risk for getting breast cancer?
From what I remember, I think it approaches nearly like 90 or 100% after.
It depends on the mutation, but on average it's around 70%. For ovarian cancer, it's around 20 to 50%. It depends on the mutation, right? I'm trying to integrate some principles of cancer genetics in these general remarks before we start. But, you know, this principle is not all mutations are the same. In terms of what can be done to prevent it, you hit on one. For breast cancer, it would be mammography or some other imaging, which would be done every year, starting when the patient is very young. Right now, the practice is only to do mammography on patients without hereditary predisposition in the general population, which accounts for 87.5% roughly of the total breast cancers. That means one in 8 people, women, will develop breast cancer during their lifetime. A small fraction of them will do so because they have inherited a mutation in BRCA1 or BRCA2. A small fraction of them, say 5% of the 12.5%. But if they have that mutation, I'll answer it in a question in a minute. If they have a mutation, you want to do screening to catch it early. The mammography is a good, reasonably good screen. It's very sensitive. For ovarian cancer, not a particularly good screening. It's with ultrasound. It's not yet been approved by the FDA for screening purposes. There is no approved screening method for pancreatic cancer. But there's something else that these women are often advised, strongly advised to do, which is not just screening. You'll undoubtedly learn about this, but they have their ovaries removed once they reach childbearing age, or once they're finished having children, because that's the only perfect way, so to speak, to preclude ovarian cancer, which is almost uniformly lethal when it occurs. Breast cancers are reasonably treatable if they're caught early. Ovarian cancers are usually not caught when they're early, and the great majority of patients with ovarian cancers die. So they're often advised to have their ovaries removed, along with a total hysterectomy, which is uterus fallopian tubes. They're often advised to have double mastectomies. And this has become routine now. Some of you may remember Angelina Jolie. She's is a famous actress who chose to have a double mastectomy. And most patients, or many patients now do, to minimize their risk. It doesn't completely avert the risk, but it reduces it to less than the risk in the general population. In the general population, it's 12%. With a double mastectomy, it's down to 1 or 2%, even in a patient with a predisposing BRCA mutation. You had a question.
I was just curious, I've heard that same number, 1 in 8 for prostate cancer. Is there some similarity between like the secondary sex organs or just how they function, or is it just
Your
chance
guess
that they're the
is as
same
good as
number?
mine. You know, no one has yet made any definitive mechanistic links between sex hormones and BRCA1 and BRCA2 mutations. So it's probably just coincidence. And prostate cancers obviously only occur in men. Breast cancers occur in men and women, although obviously much more frequently in women than men. And that gets to the last question, and we'll take this little break. Why is that? Both men and women have breast cancer. Why do breast cancers occur much more common in women than in men, both when they're hereditary or non-hereditary?
So the stab that I would take at it is that the cancer often occurs in the cells like that are producing like milk, or also just like need to be replenished, so they're growing and they're replicating a lot, and there's happening more of those in women than there are in men.
Creative, but not necessarily correct. Again, I don't know what the truth is, right? I don't know evidence to support a hypothesis. But let's finish. I'll give you a hint to finish this other. Roughly 30-40% of mutations are caused by environmental exposures. Cigarette smoke, ultraviolet light, depends on, other things that people eat, depends on the world. A few percent are caused by hereditary mutation. The rest are intrinsic, not extrinsic. Remember, every time a cell divides, 3 to 5 mutations. Okay? Cell division is a very powerful mutagen. Very powerful. It's interesting, too, that cigarettes, the way they cause increased mutations is not so much by changing one base to another, like ultraviolet light. Ultraviolet light acts at pyrimidine dimers, CC, CT, TC, and causes a change, say, of a C to an A. Right? That's a single base substitution. The way that cigarette smoke mostly increases mutation rate is indirect. It causes inflammation in the lung that makes the cells divide. And cell division is a mutagen. So the reason that breast cancers are more common in women is because there are a lot more breast epithelial cells in women. So they have divided many, many more times. And if they start out with a mutation, if it's hereditary, they will get additional mutations through this divisions, which will eventually lead to cancer. So let's take a 2-minute break, just so I can not be so hoarse and relax. If anyone wants to take a bio break.
No, I cracked another one. I hate like the way this case is designed. Like when I put my phone down, it's like the glass touches first thing.
It's not raised around the glass.
Yeah. Maybe I just need a different case. Not awake enough to answer questions.
Oh my God, I don't know.
I don't have like enough time to think about how a normal person would answer a question.
No. Well, yes. But my tax lady was like, it's not a real W-2.
Oh yeah, I gotta do our tax, my taxes. Do we still have to do taxes?
Whatever
Does the IRS like
whatever they
exist?
send you in the mail is like weird.
Oh, the thing is, they didn't know Department of Education.
Oh, is that the only thing you got?
They should send you two.
No, there's another one too, because I'm not on the health insurance and I still got like a W-2 from them.
Oh, yeah, because, okay, so there's 3 documents. There's health insurance, there is proof that they paid for your tuition, and that's just not your stipend, that's the fact that the school is paying like $30,000 so you don't have to take the classes. Then there is just your annual pay last year, which is not a W-2 because you didn't get taxes withheld. It's just a number that says you got this much money, it's
Yeah.
taxable, we haven't withheld anything.
And that's why I think my tax lady's confused, because she's like, this isn't a W-2. Okay,
It's
you
not-
still need to do something
It won't-
with
it won't-
it.
there is no W-2 for
It
your
listed as a fellowship if you're
Okay.
for her.
Okay.
She should know how to use that.
I think she does. She's just was like, I think my dad told her it was a W-2, and so she had it in her mind that it was going to be a W-2. I'm like, Dad, this isn't a W-2.
No, yeah.
Just doing it for you.
I'm like, that's why we have somebody do our taxes. Like,
But you got a form
You
that
don't-
tells you how much you got paid for 2024.
No, I
No.
had to go in.
You have to go
to
to-
my last page stuff.
Oh,
You have to go to your
gotcha.
last
Okay. Okay. Thank you.
yeah, in the
I was
corner,
so confused.
My lady in the corner will say like YTD, like year to date.
Yes.
Yeah.
I saw that. I just didn't know if I was like,
Oh,
if
yeah.
we were getting a W-2.
You like did get one.
And then Maryland state taxes, the union dues are deductible.
Oh. Nice.
Can we put this in the Slack stuff?
Sure.
That's later me stuff I gotta do, but like.
I just got that email because I sent them an email this morning and I was like, hey, I can't access my W-2.
Yesterday.
Really?
Tomorrow.
Might even be.
And they were like, oh, you don't have one. What do you mean?
I just paid someone.
Okay.
That's like I saw the
Check my email. And
Well,
I don't make enough to pay
I'm
someone.
also thinking about it because I didn't have taxes last year either. So like, they had to like backlog the file taxes for like
really simple. So I'm
3
like,
years worth of taxes. that I had.
Yeah, like I don't have
isn't the
huge
deadline?
portfolios and
Well,
I'm not too worried about. I'm not going to
15th,
itemize my deductions, so it's not.
Hopefully not.
yeah.
I don't have anything.
I don't have money in offshore accounts. Like I can
yeah, I was starting to freak out yesterday.
29 baby.
That's so many papers. Why can't they just send it all in an envelope? I know.
it's like made to confuse you. It's just like health insurance.
Way too much work.
Like what?
I got a degree in accounting. I don't
No, my friends who are accountants hate their lives. They're so bored. Like, yeah, Kyle's like an IPA. He's just like 9 to 5, no emotions. Yeah.
Don't show up.
Ready to go.
Set it. Set it at the foundation.
Brief summary of what we discussed. Perhaps the most important point is just because you read it in a journal, even a really good journal like Nature or Science or Cell, doesn't mean it's not true. Okay? And you catch the Irish in that, right? That's the most important thing I want to impart on you. The data is usually right, but the interpretation is often wrong. That's the most important advice I can give students. Because breakthroughs come when you question the interpretation. Look at the data carefully. Just because it's written there and it's past peer review doesn't mean the interpretation is right. Next thing is a question for you. I need feedback. I'm gonna give you the slide there. Anybody who wants to read through. But do you guys like this form of lecturing when I'm kind of asking you questions and challenging you, or rather than just go through the material? Good, bad?
I think it's good because it forces us to not just learn the material, but also apply the material and try and understand how it relates to each other, if that makes sense.
Good. Well, that's my hope. Anyway, that's what I'm doing. So, we'll talk about basic biology, pathogenesis, and the culprits. That means the culprit genes. I'll go through this quickly, but again, you'll have the slides. You can read the textbooks. We've written some chapters. I can pass them around. If anyone is interested, go through you. In the textbook and classic genetics. That gives the introduction. It's pretty up to date. So here's a woman with a tumor. You can see it on her forehead, right? She might think it's a beauty mark. She might call it a mole, which it's not. Technically, it's a nevus. It's benign. I'll tell you what benign is soon. Just introducing you to these diseases to make sure we're all on the same page. Here's another benign tumor. It's on the larynx. It's on the vocal cords. There's one on the bottom, two on the top. Again, it's benign, and wouldn't cause much problems, although it could alter a speaking voice, sounds. Oh, that's a huge one. Anyone knows what that is? Maya, suppose that patient came into the clinic.
I'm pretty sure it's Burkitt's lymphoma.
Oh, what?
Burkitt's lymphoma.
Oh, that's a good guess. Burkitt's lymphoma is a tumor of the lymph nodes. This is a rare tumor. It's called a hibernoma. Hibernoma, you know, you have brown fat and white fat in everybody. And we use it in humans, but bears and animals that hibernate use the brown fat. That's how they get through the winter. It's very efficient energy process. But there's some in the neck, and this poor guy had a huge one. Now, how did it grow that large? Well, he was actually on it and didn't have access to medical care, obviously, his whole life. But he finally went to a hospital. They cut it out. Here's an x-ray. And it's gone. And it's benign. Okay. Now, benign doesn't mean it doesn't kill you. There's a certain benign tumors could kill you. For example, if they're in the brain and you don't get them out. But there is a formal definition of benign tumor. The word tumor is one of the four characteristics that were originally ascribed to inflammation by the Greeks roughly 2,000 years ago. So there's pallor, heat, duller pain, rubor, redness, tumor swelling. So all of you had little, cuts or abrasions. Maybe some got infected a little bit, and they get warm and they hurt and they're red and they swell, right? And a tumor just means a swelling. That's all. So tumor types, that is swelling types, there can be non-neoplastic. So it can be a cyst that's filled with fluid, or a hernia that's filled with a segment of the bowel that comes through a little rupture in the covering. Or the ones we care about in this class are neoplastic, and there are two kinds, benign and malignant. Benign tumor, there are lots of different kinds, depends on where they occur. If they occur in an organ like the intestine, they can be called a polyp. An adenoma is a type of polyp. A nevi is a tumor of melanocytes. That woman on the first slide had it on her forehead. A malignant, another word for malignant tumor is a cancer. And there are various types of cancers. There's carcinomas, which are cancers of epithelial cells. There are sarcomas, which are cancers of fibroblast-like cells, or muscle cells, or non-epithelial cells, and then there are leukemias or lymphomas. And, whoops. Okay. Here's the difference between a benign and malignant. Now, these two tumors come from the identical cell type, a melanocyte, a pigment cell in the skin. On your left, it's a benign nevus, and on the right is a malignant melanoma. Now, what you can see is that the benign nevus, the benign tumor, has very clearly demarcated borders, whereas the malignant melanoma is spreading. It's invading. This invasion is what distinguishes the benign tumor from the malignant tumor. Now, the malignant tumor, in this case on the skin, the problem is not that it invades laterally through the skin, but that it invades three-dimensionally, that way, into the border, into a blood vessel or lymphatic, in the skin or underneath the skin. It invades in all dimensions. And once that occurs, it can escape from the skin and travel. That's what's called metastasis to a lymph node or to the lung or to the brain or to the liver or anywhere. And that's what kills patients. Metastatic cancers. The original primary tumor nowadays can almost always be cured if it hasn't metastasized. But, depending on the tumor type, in most, and about half of the patients, all cancers considered, it's not found until the last stage, until the metastatic stage, which means the tumor has been evolving for 30 years. It takes 30 years to get from a normal epithelial cell to a malignant tumor cell. So that's why one of the things we work on in our lab is on early detection, trying to find cancers before they cause symptoms or signs, when the surgeon can cure them. Yes?
Where does that number 30 years come from? Like, how is that extrapolated?
I made it up. It's true. I did make it up, but it is published in Nature, which you shouldn't believe. You should be skeptical. I made it up together with Martin Nowak, who's the chair of evolutionary biology at Harvard, in a paper we published in Nature a long time ago. And basically, it comes from knowledge of the mutation rates and the numbers of mutations that you find in cancers. So those, you know, as hard, I won't say facts, because nothing's a fact in science, but hard data. Okay, you have estimates, the number of the mutation rates, which we just went over. It's the same in cancer cells as normal cells, no different. And the number of divisions. Now, the total number of mutations depends on the number of divisions as well as the mutation rate, which is why fathers give rise to more mutations than women, right? And a corollary to that is males are responsible for most of the hereditary diseases in the world, as well as most of the other problems the world faces. That statement used to be very popular when originally these classes 20 years ago was all females. It's hardly ever we had a male in genetics course. I don't know what that was true, but everybody loved when they said that. Men are the problem. Okay, now I can't say that. Or I can, because it's true. But at any rate, so we do some hocus pocus with the math and we come up with 20 or 30 years. Right? Now, it has been substantiated since then. First of all, probably the best evidence is clinical. So certain cancers like the colon, you can track the pre-malignant lesions. the polyps, the adenomas. You can do the same with some other cancers too. And you can see when they start to occur, when they're diagnosed. And then you can see how long it is in untreated patients. Say the average age of someone getting a polyp or a benign tumor is 20, and the average age when they get a cancer is 40. So you know it took at least 20 years to get from the benign tumor to the malignant tumor, but that doesn't take into account the years that it took from that first cell which formed the benign tumor to actually multiply and become the many millions of cells that it took to diagnose it, okay? So all of these are guesses, estimates, but a reasonable guess is 20 to 30 years. But that's a good question. The Greeks, the cancer is named after the crab. Cancer means crab, I guess, in Greek. And they were looking at these invasive fingers of the neoplasm. Neoplasm just means a neoplastic tumor. Neoplastic tumor is filled with cells, not with fluids. And here's the end process. Metastasis, normal lung on the left. Metastatic cancer, say from a breast cancer on the right. Each of those nodules contains a couple of billion cells. And this is what kills patients. It destroys their lungs. Here's another example. Liver, this is a far advanced malignant tumor from the pancreas. Has metastasized the liver, it's replaced the liver with tumor. Patients can survive without livers. So this is how cancers kill in general. And again, each of those nodules, each of those metastatic lesions started with a single cell that escaped from the pancreatic cancer. 20 or 30 years after that initial cell that got the first mutation that founded the cancer, that cell traveled to the liver, through the bloodstream, lodged in the liver, and grew over a number of years, several years, so that each of those single initial cells became a billion cells. And this brings us to another number that you should remember. How often do normal cells divide? How often? Anybody want to take a guess at that? I'll give you a hint, it depends on the cell. There are some non-renewing cells, like liver, which divide not at all, basically, through the lifetime, or rarely. They're renewing tissues, like in the skin. How often do skin cells divide? Anybody take a guess? Give me a guess. Order of magnitude. Days, months, years.
A week?
Week, good guess. It's about a month. Okay? That means your cells are constantly being replaced. You are snakes. Now, the only difference between you and a snake, apart from your genes, is that snakes, they do it all at once. You know, they shed their skin all at once. But we shed our skin daily, right? So if I write on my hand like this, fortunately I don't have the ink out, but when my grandchildren paint me, it doesn't matter whether I wash it or not, they'll be gone in a week or two. Because the cells will replace, the top layer will be replaced by bottom layers, but very slowly, day by day, okay? And it completely rejuvenates once a month. In the colon, it's once a week. In the bone marrow, it depends. Red cells last for 4 months. White cells last, certain kinds of white cells, granulocytes, last for only a day or two before they're destroyed. So we have these self-renewing tissues, and there's a huge correlation between the number of cell divisions that an organ has and the incidence of cancer. And you all should know enough to understand why that's true, right? The more a cell divides, cell division is mutagenic, the more mutations it occurs that occur in that cell. And it takes a few mutations to get to a cancer, which means you need lots of divisions to get those few mutations that actually drive the cancer. And I'll talk about cancer drivers in a minute or two, but this is all linked. All right, again, there are lots of types of cancers. There are hundreds of types. Virtually every cell type in the body can form a cancer. This is sarcoma, tissue. That tumor is actually underneath the skin. It's a tumor of fibroblasts. It's a fibrosarcoma. There are also myosarcomas, which are from muscle cells. This is a tumor in the breast. What you're actually seeing here is calcification on this X-ray rather than the tumor itself. But there it is. This is a lymphoma. It's a tumor of lymphocytes. It's generally fairly systemic, but it's usually first manifest by lymph nodes in the neck. These are leukemias, tumors of the bone marrow. On your left, there's normal cells. You can see all those cells with big red nuclei on the right. They are leukemic blasts. They are the culprits. But it's the same process. They're all caused by mutations. Different numbers of mutations. In solid tumors, that is tumors of epithelial cells, most adult tumors, it takes about 3 driver gene mutations. For liquid tumors, that is leukemias, lymphomas, certain childhood tumors, it takes only about 2. So let me give you one very sad example of that. Suppose you do the most horrific experiment in history and drop an atomic bomb on Hiroshima. When do patients start to get cancer? Right side of it. Month, year, decade. Take a guess.
Decades. Like 10 years.
10 years, good guess. So they start to get liquid tumors, that is leukemias and lymphomas, about 5 years after. Why don't they get it immediately? Well, what that radioactivity did was reduce the first mutation, the founding mutation in the leukemia cell, in the cell that was eventually going to evolve to form leukemia. But that's not enough. They have to get another mutation in the progeny of that cell to get to a leukemia or lymphoma. And not only do they have to get to that second mutation, that cell has to divide enough to be recognized clinically, which means it has to become billions of cells. And we won't have time to do this today, but we can talk a little bit about the math of that. But it's not so easy for a single cell to become a billion cells, even if it's dividing exponentially every other day or so. It takes a while. If it divides every other week, it takes even longer. So you start to see them 5 years after dropping of the bomb. or after Chernobyl accident. Or it used to be, even before I was born, that when you went to get a pair of shoes, that they used to x-ray your feet to see what your bone structure was. That was before it was realized that x-rays are mutagens, which could result in cancers. That was not a good idea. Definitely one of the worst ideas it is a good idea. I mean, it seemed like a good idea at the time. So, you could walk in your neighborhood shoe store and get an x-ray. And those patients, often children, occasionally would get cancer. It wasn't as bad as I'm making out to be, but it was certainly not a good idea. Anyway, these people, these poor people who survived the atomic blast, they didn't start to get cancers of the colon or breast till 20 or 30 years after. Which is another answer to your question, Carter, about how long it takes to get a cancer. Probably a better and more statistically valid answer than what Martin Novak and I did. A lot of 40-year-old models. Okay, so where are we? Here. Now we'll talk about in the last. Okay, finish with this, then we'll start on Wednesday. So, cancer is a genetic disease, but it's different from all the other genetic diseases, or almost all that you'll discuss in this course. Take a typical genetic disease, monogenetic disease, Huntington's disease. A patient who has inherited a mutation in the Huntington disease gene, they will get Huntington's disease. They will get the same symptoms, same severity. Doesn't matter whether they grow up in smog-filled Los Angeles or serene Sri Lanka, they will get it. Okay, because it has no environmental component, none at all, and there's no treatment. So this is purely genetic. We talk about nature and nurture since the time of Darwin. This is nature. It's genetic. That is many of the diseases you'll talk about in this course, but far more common are diseases which are the result of the combination of nature and nurture, of genes and environment, contributed to by both. An example is LDL receptor, lipoprotein receptor, low-density lipoprotein receptor. You guys have all talked about this disease in the course already. So, no? Okay. Well, this is, again, a cause of atherosclerosis, just like cancer. It's not responsible for most atherosclerosis. In this case, what's responsible for most atherosclerosis is not genetic. But even in those patients who inherit a mutation in the low-density lipoprotein receptor, they won't necessarily get atherosclerosis. It contributes, that mutation contributes, but it's not sufficient. It doesn't cause it, it contributes it. And the other contributor here is diet, particularly lipids. And the combination of diet plus the mutation makes atherosclerosis, and this mutation predisposes them to the disease. So patients who have the same diet, if they also have an LDLR deficiency, genetic, they're more likely to get atherosclerosis. That's the difference between predisposition and prevalence or incidence. And with cancer, it's the same. There are certain patients who inherit a predisposition to cancer. They won't necessarily get cancer. It depends partly on the environment, but partly on just bad luck, whether they happen to get a mutation through the natural course of the cause of most mutations, which is cell division. So as I mentioned before, patients who have a BRCA mutation, they may have a 70% chance of developing ovarian cancer and a 40%, 70% of breast cancer and 40% of ovarian cancer. But in all of these cases, and in breast and ovarian in particular, it's not so much environmental agents that cause these other mutations that are responsible for these other mutations. It's just the mutations associated with cell division. So again, it's bad luck. Now, in colon cancer, that's another example. 95% of, well, let's say 85% of patients who develop colon cancer do not have any hereditary predisposition. They have no mutation in their genome, in the germline, that predisposes them. All of those mutations come from the other sources of mutations, which is environment and bad luck, which is cell division. Now, that doesn't mean you can't reduce your chances of cancer by avoiding environmental causes of cancer, which of colon cancer, and that is a very active area of research. You can't get rid of the naturally occurring mutations that occur with cell division. That's impossible. But you can get rid of mutations that are associated with environmental causes. And there have been numerous environmental associations, correlations over the last couple decades, starting with meat was bad, starting with cooked meat, or continuing with cooked meat. There's some evidence that fiber-based diets high in fiber can protect you by protecting the intestinal lining from more divisions. There are a whole host of environmental factors, particularly dietary, that have been shown to raise the incidence of colon cancer, but the exact particulars of the diet that are responsible for this are still an active area of debate amongst epidemiologists. And from a molecular genetics perspective, one of the great unsolved mysteries is if one assumes that these environmental influences, food is important, how do those foods cause mutations? For any of those of you who have an epidemiological bent and are interested in an important question, that's a good one. And it's the same for many other cancers. So that's what I'll leave you with. We'll go through these numbers when we start on Wednesday. And if any of you have any questions in between now and then, save them up. We'll start with them. Thanks. See you Wednesday.
Thank you.
They classified like bacon as a carcinogen, but like the risk is so low. It's like if you live in a city, it's probably equivalent like the smog that you would consume, and like stop attacking bacon.
Yeah, I did that.
Look.